I'm going to move that Instapol. It's not showing. About that. Well,
That's weird.
maybe I have to skip this first. All right, now you can see what I'm seeing. Stop me if you don't see what I'm seeing.
Well, they didn't know what you were
That's
saying.
true. So this button, I'm going to move to the top so it's easily accessible. And I ask you all to, I'm going to ask you a couple of more questions. There's no right or wrong answer, so this is about participation. And we know that electronics fail sometimes, so we're going to most frequently ask more than one question. So if one of your questions didn't go through and one did, that's all you need to have
For full
being
participation points that day.
registered
Yeah.
that day. All right?
Wrong answers count, strictly participation.
So we just asked you this question. I have 59 responses. Anybody's trying to log in? Okay. Okay. Be sure to log in and then you'll be able to get your response for the next one. Let's see where we are at. So you all are talking. Interesting. The biggest thing is organelles and mitochondria. All right. I like microscopic, life, membrane, communication, pathways. Yes, we're going to cover pathways, cycles, maybe cell cycle, chemistry a little bit. Microbiology, communicate, yes. Complicated, not so complicated.
Pretty complicated, I think.
I don't see fun.
It's fun. Complication can be fun.
Okay.
Mitochondria always comes up. This is the first year that organelles have been equal to mitochondria. Yay. And is your sound up?
No. Okay, so how about that? Can you hear me well? More? Okay, so, ooh, too loud. All right, let's try that. Great. Okay, so, awesome. I'm going to ask you a couple more questions in just a minute. So, so let's get into the matter. So we are going to be talking about cell biology. So, but let me be clarified. Cell biology is a huge, gigantic field. So we are going to be narrowing that field to just focusing on animal cell biology. So we're not going to be covering, unfortunately, don't have time to cover plant biology. We don't have to cover, there's a lot of really interesting biology of bacteria that is not covered as part of microbiology, but again, there's just way too much to learn. So we are going to try to cover some topics of the biology of eukaryotic cells, and we are going to try to choose a few topics and go quite deep beyond the contents of what is covered in your textbook. So our goal is for you to understand not only the parts of the cell, which probably you have been hearing from previous courses, introductory courses. Yes, we're going to go through some of those, some of the basics, but really the important aspect is to understand how is it that the cell works, how does the cell use its proteins to achieve the jobs that they need to accomplish. And very importantly, how do we as scientists do experiments to figure out how the cell works. So we're going to be doing a lot of, thinking a lot about the cell from an experimental point of view to see how is it that the contents of what you see read in the textbook was initially found, so that then you are prepared for all the new information that will be discovered, that is currently being discovered and will be discovered later down the road. You will have the tools to analyze that. So we are going to try to go into the molecular side of cell biology, so kind of combining biochemistry with cell biology. So we're really going to try to look at the components of the cell and how they are put together to accomplish the function of the cell. And as you probably have seen in previous courses, a bulk of the contents of the cell is constituted by proteins. So proteins really are the workhorse of the cell. They are the ones that are going to be doing the job of the cell, making an organelle function, transporting things around. All of those are going to be accomplished by proteins. So 90% of the content of this course is going to be all about proteins. And therefore, we really need to think carefully about how we study proteins and how we can use experiments to figure out how those proteins work in the cell. And yeah, we're going to cover a little bit of polysaccharides and other components. We're not really going to talk much about DNA and RNA beyond being substrates for proteins during replication. All right. So since we're going to be talking about proteins, it's going to be essential that you go back and read and memorize all the 20 amino acids and their properties, so that we have a language in common to talk and move forward. So we want you to go back to the textbook. We also created a PowerPoint that has everything we want you to know about the amino acids. So you need to know the names of the amino acids and their 3-letter code and 1-letter code, because we're going to be using that constantly in the course. So you need to be very familiar with them. And then what are their general chemical properties? We will not ask you to draw the chemical structure of them. So you don't have to actually be able to draw them, don't memorize that. But in that PowerPoint slide that is posted on Canvas, we do have some discussion about chemical properties that are important beyond just generally what is it positively charged, uncharged, or hydrophobic. That is important, but we want you to know more beyond that. For example, there's a slide that tells you that serine and threonine and tyrosine have a hydroxyl group, an OH group, that can be modified in a variety of ways. For example, it's a great substrate for attaching a phosphate through phosphorylation. And we're going to talk a lot about phosphorylation in this course, so that's going to be really important to get to know. And why? Because it has a hydroxyl group. That's all you need to know about those guys. And like that comes a couple other elements for the other amino acids.
But the properties of the side chains can change according to pH and whatnot. Some of them are sort of indeterminate. So we're going to agree to use these properties as is in this Albert's textbook. So
And the PowerPoint,
arginine
and the PowerPoint slide.
and the and the PowerPoint slide. So arginine will always be histidine will always be considered positively charged, for example, and the nonpolar ones are not always nonpolar. So you can look at that for the general properties, and then that we'll agree this is what we're going to use, even though in the test tube properties might change. And then as well as the additional properties that we'll talk about that are specific for some of these, like phosphorylation and ubiquitination and things like that. So we'll be covering in the class. Any questions?
So a quick review of the structure, how the structure of a protein. So each individual protein is going to have a unique amino acid sequence. It's a primary structure, right? And then that primary structure is going to determine how the protein is going to fold first into a secondary structure, like alpha helix, beta strands, and then it's eventually going to fold into a 3-dimensional structure that is going to be the tertiary structure of the protein. So all of that, we're hoping that you come in with an understanding from previous coursework. So in the final folded protein, the surface of the protein is going to be enriched in hydrophilic amino acids that can interact with water. In the core of the protein, buried inside the protein, we're going to have an enrichment, not 100%, but enrichment of hydrophobic amino acid side chains, right? So, anybody has run an SDS gel? SDS acrylamide gel, all right, a lot of people, all right. So the SDS polyacrylamide gel electrophoresis is going to be the workhorse of how we are going to be studying proteins. It's the one of the main methods currently still used in all laboratories that undergo studies of protein function. So I think it's a good thing to just quickly review how SDS gels work. Essentially what you do is you make a gel, a slab of gelatinous material that is as a polymer of acrylamide, so that's why it's polyacrylamide. It acts as a sieve, so it's porous, and proteins can navigate or flow through that gel. To make them move through the gel, we're going to apply a current through the gel to then make the proteins move by charge. So we are going to turn on the current. We have the positive electrode down at the bottom, and the proteins are going to be loaded on wells at the top, and then the proteins are going to migrate through this gel by the action of the current. But as you know, proteins have a variety of amino acids, and some of them are going to have a lot of positive amino acids, some of them are going to have a lot of negative amino acids, and some balance in between. So the rate at which they move through this gel could potentially depend on the charge of their amino acid side chains. So if we have a protein, for example, that had a lot of positive amino acids, it's not going to want to move easily towards the positive electrode. So to try to standardize the movement of proteins through the gel and make them move by the relative size, we use a trick, which is to denature them with a very strong detergent called sodium diallysyl sulfate, or SDS, shown in this line. So this is a strong ionic detergent that has a sodium ion. It has a really long hydrophobic side chain there. It's not amino acid, so it's an aliphatic chain there that is going to interact with proteins and is going to coat them essentially all along their length of the protein. By the action of the detergent, and also because we're going to boil the protein sample, we're going to cause the denaturation of the protein from the folded structure into a linear unfolded state. So the protein goes from having its own charge to now being coated by a very large amount of detergent. All of the molecules of the detergent are negatively charged. So essentially, we have kind of negated the intrinsic charge of the protein, and now we have made it all uniformly negatively charged. So now all proteins in the protein sample, independent of their amino acid composition, more or less, will be able to migrate through the gel, attracted towards a positive electrode when we apply the charge. And they are going to be separated by size because they have to move through this barrier of the gel, which basically is this gelatinous material that has holes through which the proteins have to wiggle through. A protein that is small, that is short, will be able to move through the gel very fast, wiggle quickly. A protein that is very large is going to get entangled some more, so it's going to migrate slower. So what we obtain is the separation of proteins by size. Small proteins run faster, big proteins run slower. All right? Questions? Good? Everybody